Good morning, students. Today we shall discuss about antigens. An antigen is a substance that is recognized by the immune system and can trigger an immune response. When an antigen enters the body, it stimulates immune cells such as B lymphocytes and T lymphocytes to produce antibodies or activate cellular immune responses. The term antigen is derived from antibody generating substance. Most antigens are proteins or polysaccharides present on the surface of microorganisms such as bacteria, viruses, fungi, and parasites. The key characteristics of an antigen is its specificity, which speaks about antibodies produced in response to a specific antigen only bind with that antigen or very closely related ones alone. In the aspect of non-covalent bonding, the binding involves weak, reversible forces including hydrogen bonds, ionic bonds, hydrophobic interactions, and van der Waals interactions. Lock and key model. The strength of the binding is between the antigen and antibody. It is like a lock and key model. It binds very close to each other. The structure of an antigen and epitope. Antigen contains specific regions called epitope, or it is also called as antigenic determinants. Epitope. The epitope is the specific part of an antigen that binds to an antibody or immune receptor. Antibodies do not bind the entire antigen, they bind only to these small regions. An epitope usually consists of 5 to 15 amino acids or a very few sugar residues. Paratope. The paratope is the antigen binding site on the antibody. The epitope and paratope interact through non-covalent bonds such as hydrogen bonds, electrostatic interactions, and hydrophobic interactions. A single antigen may contain many epitopes, allowing different antibodies to attach for the different parts of the antibody at the same antigen. Characteristic of an antigen. For a substance to act as an antigen, it usually has the following properties. The foreignness. The immune system recognizes the foreign part that is nothing as the antigen. High molecular weight. The large molecules that is usually about 10,000 Dalton are better antigens. Chemical complexity. Complexity molecules such as proteins and polysaccharides are strong antigens, while simple molecules are weak. Specificity. Each antigen has unique epitope, allowing antibodies to recognize it. The ability of an antigen to induce an immune response is called as immunogenicity. Types of antigens. The different types of antigens are exogenous antigens. They enter to the body from outside. Example, bacteria, viruses, pollen, dust. Secondly, endogenous antigens. They are produced inside the infected cells. Example, viral proteins are synthesized inside the host cells. Thirdly, autoantigens, normally body components that become targets of the immune system, seen in autoimmune diseases. Fourthly, tumor antigens, markers on the surface of tumor cells often caused by mutations that alert the immune system. Fifthly, complete antigens can independently stimulate an immune response. Sixthly, haptens, small molecules that cannot induce an immune response alone, become antigenic only when they attach to a carrier protein. Antigen-antibody reactions. When an antigen enters the body, antibodies bind specifically to its epitopes, forming an antigen-antibody complex. Steps that are involved in antigen-antibody reactions are recognition, binding, formation of antigen-antibody complex, then the response, which leads to neutralization of toxins, agglutination of microbes, precipitation of antigens, phagocytosis, and destruction of pathogens. These reactions form the basis of humoral immunity. Antigen and interactions. When antigen-antibody interaction is a specific chemical reaction that occurs when an antibody binds the antigen that stimulates its production. This binding is highly specific and forms the basis of immune defense, diagnostics tests, and vaccine responses. Antibodies recognize specific regions of antigens called epitope and bind to them using their antigen binding site, that is nothing else, the paratope. Antigen and interactions are highly specific, reversible binding events between an antigen's epitope and antibody's paratope, serving as a cornerstone of immune defense. These non-covalent interactions facilitate pathogen destruction and are widely used in diagnosis. Nature of an antigen antibody binding. The binding between antigen antibody is non-covalent and reversible. It involves weaker intermolecular forces such as hydrogen bonds, electrostatic interactions, interactions, van der Waals forces. These interactions together create a stable antigen-antibody complex. Features of antigen-antibody interactions. Specificity. Each antibody binds only to the specific antigen determinant, that is epitope, that stimulates its production. Affinity means it refers to the strength of binding between a single antigenic epitope and an antibody binding site. Avidity speaks about overall strength of binding between an antigen and multiple antibody binding sites. Reversibility means since the bonds are weak, antigen-antibody interactions are reversible and certain conditions such as it changes due to its temperature or pH value. Stages of antigen-antibody interaction. In the antibody recognizes the specific antigen through its epitope. It leads to binding. The antigen binds to the antibody through the antigen binding site, followed by formation of antigen-antibody complex. Stable complexes are formed, and the biological response speaks about the immune system eliminates the antigen through various mechanisms. Outcomes of antigen-antibody reactions. Neutralization. Antibody neutralizes toxins or viruses by blocking their harmful effects. Agglutination. Antibodies cause clumping of cells or microorganisms, making them easier to remove. Precipitation. Soluble antigens combine with antibodies to form insoluble complexes. Opsonization. Antibody caught pathogens and make them easier for phagocytes to engulf and destroy. Complement activation means antigen-antibody complexes can activate the complement system, leading to the destruction of the pathogens. Factors that affect the antigen-antibody complexes are temperature, pH, concentration of antigen-antibody, time of reaction, presence of electrolytes. Optimal condition increases the efficiency of the reactions. Importance of antigen-antibody interaction. It provides immunity against infections, basis for vaccination, used in diagnostic tests such as ELISA and blood grouping, important immunological research, and it helps in disease detection. Role of antigens in the immune response. Antigen play a very crucial role in maintaining or in initiating the immunity. It stimulates B cells to produce antibodies. It also activates T cells through antigen presentation. This also helps the body to identify pathogens and infected cells. It also provides the basis for vaccination and immunological memory. Antigens are often processed by antigen-presenting cells such as macrophages and dendritic cells, which present antigen fragment to T cells to initiate the immune responses. Examples of antigens are bacterial cell wall proteins, viral envelope proteins, pollen grains, toxins produced by bacteria, and foreign blood group antigens. Conclusions. Antigens are fundamental components in immunology because they initiate, regulate immune responses. Their interaction with antibodies and immune cells form the basis of host defense, vaccine development, diagnostic tests, and immunotherapy. Antigen, it stimulates the immune response. Antibody, it is produced in response to an antigen and binds specifically it. Comparison of antigen and antibody in definition means antigen, a foreign substance that stimulates an immune response. Antibody, a specific protein produced by B lymphocytes in response to an antigen. The nature of an antigen, it is usually protein, polysaccharides, lipids or nucleic acids. The nature of the antibody is they are glycoproteins belonging to the immunoglobulin family. The function of the antigen is it triggers the immune system to produce antibodies and activate immune cells. The function of the antibody is to recognize and bind specifically to antigens to neutralize them. The source of antigens, they are bacteria, viruses, fungi, parasites or other foreign substances. The source of antibody are B cells and T cells which are produced from thymus and bone marrow. The specific region of antigen is epitope or antigenic determinant recognized by antibody. And the specific region of antibody is a paratope or antigen binding site that bind to an epitope. The location found on the surface of microorganism or foreign particles. The location of antibodies, they are found in the blood plasma, lymph and body fluids. The role of in immunity, it initiates immune response. It provide in the case of antibodies, it provides defense by neutralizing or destroying the antigens. The examples of antigens are bacterial toxins, viral code proteins, pollen grains. The examples of antibodies are immunoglobulins, IgG, IgA, IgE, IgD, and IgA. Thank you.